Alright, this video lesson is about prokaryotes, eukaryotes, and the endosymbiotic theory. So pay close attention, record your notes down in your journal, and write down everything that's on the screen and anything that I might add to the screen. Make sure that your notes are complete in case I give you a notes quiz over this video. Also, make sure that you pause if you need to pause the video and rewind and rewatch parts that you missed. Alright, so let's get started. Right, first we're going to begin with the prokaryotes, aka bacteria. So prokaryotic cells, whether you are aware of it or not, are actually all around us. Okay, they are on your skin, they're on your eyelashes, they live inside of your intestines to help you digest your food. They are on your pencils, they are everywhere. In fact, there are more prokaryotic cells on and in your body than you have of your actual own cells. So you are more of a bacteria than you are actually a human. It's kind of crazy if you think about it. So bacteria have been around for a really long time. In fact, they are considered to be the oldest living cells on Earth. Now, Earth is thought to be about 4.6 billion years old, right? So it's really, really old. And the first prokaryote is thought to have arrived about 3.5 billion years ago. So these are the oldest living cells on Earth, and we can go back and look at fossilized structures from this time period, and scientists can do all different types of dating to figure out the age of these. And so prokaryotes are really old, and they have not changed much over that time period because they're just really good at being single-celled organisms, so they haven't needed to evolve. Some characteristics of prokaryotes. The main characteristic you need to make sure you know, and I want you to highlight this, I want you to box it, is that they have no nucleus, right? I tell my students to remember this because pro equals no, okay? So prokaryote equals no nucleus, right? That is one of the distinguishing factors that easily separates it from a eukaryotic cell. Okay, another distinguishing factor is that it has no membrane-bound organelles. This means it does not have any mitochondria, it has no endoplasmic reticulum, no lysosomes, no nucleus, no vacuoles, none of that. Okay, so no membrane-bound organelles. They always have a cell wall. Okay, and this cell wall is made up of a protein and sugar called peptidoglycan. Right, and peptidoglycan is actually very important in the structure of the cell wall, and without it, the bacterial cell can't actually make its cell wall, and we'll talk about why that's important in just a minute. Some other things they always have is they're always going to have a cell membrane. They always have DNA, that's a requirement of all living things. Remember, all living cells have to have DNA or genetic material. Right? They always have a cytoplasm. Remember that cytoplasm is that gel-like fluid inside of the cell. And they always have ribosomes, okay? Because ribosomes are used to make protein. So no matter what type of cell you are, whether you are a single-celled organism like a prokaryote or you are a part of a multicellular organism, all cells have to be able to make proteins in order for them to be able to function correctly. So all cells make protein using ribosomes. Reproduction. Prokaryotes reproduce asexually most of the time through a process called binary fission, right? And this is asexual. In this process, we start with one bacterial cell. It grows a little bit bigger, and then starts to kind of divide in half, and then eventually we get two bacterial cells that are identical to the parent. Okay, so it's just dividing the cell in half, right? So that is asexual. The offspring are identical to the parent. Sometimes it can do kind of sexual reproduction by exchanging genetic material through a pilus during what we call conjugation. Okay, so here is one bacteria, and here is another bacteria. They have this DNA inside of them like this. They can create a little tube called a pilus between the two of them, and the DNA can be exchanged back and forth. Okay, so during this process, they are exchanging genetic material, so we do get a recombination of the genes in the bacterial cell, But this is not true sexual reproduction because we don't have two parent gametes coming together. So it would still be asexual, but we are getting some different combinations of the genes. And this is going to be important for things like antibiotic resistance, because this is how it happens, right? It happens through mutation, but also through bacteria exchanging that genetic material with each other and passing along those genes for being resistant to different antibiotics. So that is something right now that's on the rise as we use more and more antibiotics and antimicrobial things, is that bacteria are becoming resistant, more and more resistant to those things as we use them more often. All right, so to get energy and obtain energy and use energy, we have two different categories: autotrophs, which are able to make their own food. Example of an autotroph is a cyanobacteria, which is actually able to perform photosynthesis, okay, just like a plant. So it uses the sunlight to make its own food. And heterotrophs, this is what most bacteria fall under. They're mostly heterotrophs in that they consume other organisms or byproducts of another organism. So for example, E. coli is actually a bacteria that lives in our intestines and helps with our digestion. Okay, so this is important in helping us to break down the food that we didn't get to finish breaking down all the way. And so if you've ever taken an antibiotic, you're probably aware that sometimes it can make your tummy upset and make you have diarrhea because it has disturbed the colonies of the naturally occurring bacteria in your intestines. It has killed those, and so now your body's not able to digest your food as well. That's why sometimes people recommend eating the yogurts and stuff with the live cultures in it to help restore your balance in your digestive tract. So bacteria can help us with some things, as we'll discover in a minute. So benefits of bacteria, like I just said, are helping with digestion, helping us to break down our food, and we also use them to make cheese and yogurt and other food items that we all like to enjoy. So even though they aren't necessarily our favorite thing, and sometimes we think they're gross, they actually do have a lot of benefits for us, even though they do get a bad rap. Now, there are some downsides to bacteria, and I'm sure all of you have probably been sick in some form or fashion, and it's probably been caused by a bacteria. If you've ever had strep throat, that would be caused by a bacteria. And to treat bacterial infections, doctors often prescribe antibiotics. Now, antibiotics are only used to treat bacterial infections, okay? They will not work on a virus, okay, like the cold virus or the flu virus. They only work on bacteria. Okay, and the way they work is that they actually prevent the bacteria from linking together that peptidoglycan in their cell wall. So if it can't form a cell wall, then the bacteria is going to burst and it's not going to be able to live. So it's not going to be able to protect itself from its environment. Okay, some main differences between bacteria are whether they are gram-positive or gram-negative. Gram-positive bacteria are going to stain a purple color because they're going to absorb more of the dye because they have a thick cell wall. So if you look down here at my picture, you can see that these circle ones here, those are going to be our gram-positive bacteria because they are the purple-looking ones. Gram-negative are going to actually stain pink because they have a thin cell wall, so they're not going to absorb as much of the stain. So if we look here, that one would be a gram-negative because it is not absorbing much of the stain and it's going to be pink. It's important that you know the difference between those two. We use those, scientists use those to help design antibiotics because depending on whether it is gram-positive or gram-negative determines whether or not the bacteria will be able to work with different antibiotics. All right, so now we're going to get our drawing caps on. We're going to draw a prokaryotic cell. So here we go. The first step is we're going to draw, it looks kind of like a pill. So here's our prokaryote, here's the outside. This is going to be our cell wall. And then we're going to draw another line here, which is going to represent our cell membrane. So remember, they always have a cell wall. So here's our cell wall. And this inner line would be the cell membrane. Right, over here, we're going to draw some little fuzzy things. These are called pili. Okay, and these are what are going to help the prokaryote to fill around in its environment and move around its environment. On the inside fluid here is called the cytoplasm. In here we're going to have a region, this is our nucleoid region, where it doesn't have a nucleus, but it has its DNA concentrated in the center region of the cell. And so this here would be the DNA of it. And these dots here inside are the ribosomes. So those dots are the ribosomes, and those are responsible for making the proteins for the cell. So take a moment, pause, make sure you have the structure drawn, because I will expect you to be able to label the structure of a prokaryote. Right, now that we've talked about prokaryotes, let's move on to eukaryotes. Eukaryotes, we're actually going to spend all of Unit 6 talking about all about eukaryotic cells and their organelles. So this is going to just be a brief introduction into those. These are complex cells. Unlike prokaryotes, which are considered to be simple cells, these are extremely complex. Okay, and these are the types of cells that make up multicellular organisms like animals and plants. Some things about eukaryotes that make them stand apart is that they always have a nucleus. Okay, so remember I told you about prokaryotes was pro equals no. Well, you can remember U equals do, and that they do have a nucleus. Unlike prokaryotes, they always have membrane-bound organelles. Okay, and we're going to talk a lot about these in Unit 6. They always have a cell membrane, cytoplasm, DNA, ribosomes, and unlike bacteria who always have a cell wall, eukaryotic cells sometimes have a cell wall. Okay, and this is going to depend on whether we're talking about an animal cell or a plant cell or a fungal cell or a protist. Okay, because animals are actually not going to have cell walls, and that's one of the reasons why animals are able to be so mobile and flexible and have such a wide range of movement like we do. So eukaryotic cells are used to make up protists, which are actually going to be unicellular for the most part, okay? Plants, animals, and fungi, okay? And so, like I said, we're going to spend all of Unit 6 just talking all about these eukaryotic cells. So lastly, we're going to talk about the endosymbiotic theory. All right, and this is a theory that scientists use to help explain how we went from prokaryotic cells 3.5 billion years ago to getting eukaryotic cells later after that, about 2 billion years ago. So the endosymbiotic theory simply states that eukaryotic cells arose from prokaryotes, and this is how it says it's done. Okay, so we first have a large prokaryote which ingests or eats a small prokaryote. Okay, so I kind of think of this as like a big bad bully picking on a small little prokaryote. So big bad bully, large prokaryote comes along and eats a small prokaryote, picking on him, okay? So then the second step is that the small prokaryote is trying to make a deal, okay, because they're like, well, I don't really feel like being eaten today. Is there anything we can do to kind of work this out? Is there anything I can do for you? And maybe, you can protect me. What kind of deal can we work out? So the small prokaryote agrees to produce energy for the large prokaryote, okay, and give him extra energy. So the large prokaryote can just go on and live its life and not have to worry about making as much energy, since the small prokaryote will be doing it for him, and vice versa. The small prokaryote now is protected from other large prokaryotes that might want to eat him. So eventually, this deal works out so well that the two cannot live without each other. And so they live together in what we call a symbiotic relationship, where they become dependent upon each other. So over time, the small prokaryote turns into either the mitochondria or the chloroplast. All right, so here's an illustration of this. You can see here's our large prokaryote in the first picture here, ingesting the small prokaryote. Eventually, they become either the mitochondria, and that becomes an animal cell. If it also engulfs some cyanobacteria, then it's going to become a plant cell. So here's some evidence that supports this. The mitochondria and chloroplast actually have their own DNA that's different from the DNA in the nucleus of the cell. So even in your cells, in your body, your mitochondrial DNA comes from your mother, okay? And it's different than the DNA that's found in the nucleus of the cell, right? In addition, both mitochondria and chloroplasts have a structure that resembles the prokaryote. So mitochondria kind of look like this here, which resembles that of a chloroplast. And with a prokaryote, same with a chloroplast, has this structure here. So they both kind of look similar to that prokaryote that we drew earlier. And lastly, both mitochondria and chloroplasts are used to provide energy for the cell, just like the endosymbiotic theory states that they lived in harmony because the small prokaryote provided energy for the large prokaryote. All right, so this is the conclusion of our notes. So make sure you have everything written down and rewatch any parts that you need to and be prepared for a quiz should I give you one. All right, have a good evening.